Welcome back to the deep dive. Today, we're taking a close look, a real structural inspection, if you will, of maybe the most essential boundary in all of biology, the cell membrane.
Yeah, it's fundamental.
It's so much more than just a container, isn't it? It's this incredibly dynamic, actively managed frontier that really defines what a cell is and, what it talks to.
Absolutely. It's the cell's master organizer.
Yeah.
I mean, the sources really highlight its first job, defining inside from outside, creating those compartments.
Right.
But the really mind-blowing part is its role as like a bouncer, a master regulator controlling everything that goes in or out. That's selective permeability.
Okay, selective permeability. Let's unpack that system because the membrane, it seems like it's built from just a few key things, right? We've got the basic structure, the lipids and cholesterol.
The foundation.
And you have the really complex functions, mostly run by proteins.
Yeah, the machinery.
And finally, the transport systems that actually let the cell interact and communicate. So 3 core components to explore.
That's a good way to break it down.
So let's start right at that foundation. The phospholipid bilayer. You look at these molecules, and it almost seems like magic how they just form a wall in water.
Well, it feels like magic, but it's pure chemistry and physics, really.
Yeah.
The key thing to grasp is the energetics. Phospholipids, they're sort of two-faced molecules.
Okay.
You have a phosphate head part that loves water, hydrophilic, and then these 2 fatty acid tails that absolutely hate water, hydrophobic.
Right. Oil and water don't mix.
Exactly.
Yeah.
So when you dump these things in water, they don't need instructions. The tails desperately try to get away from the water, so they tuck inwards, facing each other.
Okay.
And the water-loving heads naturally face outwards towards the watery environment inside and outside the cell. Boom, you get a bilayer. It's the lowest energy state, totally spontaneous, self-assembling, even self-repairing.
A wall built out of necessity, then. Okay. But just lipids. That sounds kind of flimsy, like it might fall apart if things get too hot or cold.
That's where cholesterol comes in.
Yeah.
And, gets a bad rap in diets, but in the membrane.
Yeah.
It's absolutely essential. It's the structural backbone, the stabilizer.
No, it makes it tougher.
Tougher, yes, but also more adaptable. It acts like a fluidity buffer. When it's cold, phospholipids want to pack tightly together and get brittle. Cholesterol wedges in between them, keeping them spaced out, maintaining fluidity.
Okay, prevents freezing up.
Right. And when it gets hot, lipids want to spread apart, making the membrane leaky. Cholesterol kind of pulls them together, preventing excessive fluidity and weakness.
So it works at both extremes.
Precisely. But cholesterol's role is even more sophisticated now we understand lipid rafts.
Lipid raft. Okay, what are those? Sounds interesting.
They're these little microdomains within the membrane. Think of them as slightly thicker, less fluid patches that are really rich in cholesterol and specific lipids.
Like little islands in the membrane sea.
Kind of, yeah. And they act as organizing centers. The cell uses them to cluster certain proteins together, maybe signaling receptors or adhesion molecules.
So instead of things just floating around randomly.
Exactly. It concentrates the necessary components for a specific task, like triggering a signal pathway, all in one place. It makes the cell way more efficient, like having dedicated workstations.
That really elevates it from just a barrier to an organized platform. Okay, so we have this durable, organized foundation. Now let's get to that main job, deciding who gets in and who stays out. Selective permeability. What are the basic rules?
The rules are pretty strict and mostly boil down to 3 things. Size, polarity, meaning is it water-loving or fat-loving, and electrical charge.
Right.
Generally, the smaller and more non-polar or fat-loving a molecule is, the easier time it has crossing that fatty lipid core.
So top of the list, fastest passage,
Yeah.
small non-polar stuff like gases.
Exactly. Oxygen, carbon dioxide,
I
they
think
barely even notice the barrier. They just diffuse right through down their concentration gradient. Easy.
Okay. What about something small but polar, like water? It likes water. The core hates water.
Good question. Water can actually slip through, but it's slow. Despite the hydrophobic core being a deterrent, water's just so small and abundant, some molecules manage to sneak past occasionally. Slowly, though. And bigger nonpolar things. Right. Fat-soluble vitamins. Vitamin A.
Right, retinol, yeah. Vitamin A1. It's nonpolar, so it likes the lipid core, but it's relatively large, so it crosses, but again, slowly. Size becomes a limiting factor there.
Okay. Now for the really important stuff the cell needs constantly. Glucose, amino acids. They tend to be larger and polar, right?
They are. And that's where the membrane draws a hard line. Large, polar molecules like glucose. Blocked. The combination of size and their preference for water means the fatty core is a major barrier.
And ions. Sodium, potassium, calcium, essential for nerves and muscles. their charge.
Zero chance. Absolutely no chance of passing through on their own. That electrical charge interacting with the nonpolar lipid environment creates a massive energy barrier. They are completely repelled.
So the membrane is naturally impermeable to many of the most vital things the cell needs. Precisely. And that's why the cell had to evolve all this amazing protein machinery we're about to talk about. The impermeability forces the need for specialized transport.
Right, this is where it gets really dynamic. The things that can't just diffuse need help. So the cell embeds proteins into the membrane. And this whole picture, lipids, cholesterol, floating proteins, that's the fluid mosaic model, isn't it?
That's the one. It emphasizes that the membrane isn't static. It's fluid, with proteins and lipids able to move around laterally, like tiles in a mosaic, but ones that can shift position.
And these proteins are the workhorses. You mentioned different structural types, integral, peripheral.
Yeah, structurally you have integral ones that go all the way through, peripheral ones sitting on the surface, and some lipid-bound ones tucked within a layer. But functionally, the transport proteins are key for getting things across.
Okay, the traffic controllers. Let's break down the main types. First up, simple tunnels, channels.
Channels are basically protein-lined pores. They form tunnels that allow specific things, usually water, like through aquaporin channels or certain ions, to pass through rapidly down their gradient. They can be gated, opening or closing, but it's still passive flow.
Like an open door, basically. Then you have carriers. How are they different?
Carriers are much more specific and complex in their action. They don't form an open tunnel. Instead, they have a specific binding site for their target molecule.
Oh.
Binding causes the carrier protein to change its shape, kind of like a revolving door with sequential gates. It opens to one side, binds the molecule, closes that side, then opens to the other side to release it.
And the GLUT4 glucose transporter is the classic example here. Walk us through that gate system.
Sure. GLUT4 starts open to the outside of the cell. Only glucose fits its binding site. When glucose binds, that triggers a conformational change. The outside gate effectively closes.
Seals it in.
Seals it in. Then the protein shifts, opening an inner gate towards the cytoplasm. Glucose is released inside the cell, and the transporter then resets back to its original state, ready for another molecule.
Very precise. Ensures only glucose gets in that way.
Exactly. High specificity prevents leaks.
Okay, channels and carriers mostly handle passive movement down the gradient. But what about moving things against the flow? That takes energy, right? That's where ATPases come in, the pumps.
Yes, the active pumps. These are enzymes, ATPases, that harness the energy released from breaking down ATP.
The cell's energy currency.
Right. They use that energy to actively pump molecules or ions against their concentration gradient, from low concentration to high. It's like pushing something uphill. It requires work.
And the prime example is the sodium-potassium pump, the Na plus K plus ATPase. It pumps 3 sodiums out, 2 potassiums in. Why is that specific ratio so important?
It's incredibly important because it's moving more positive charge out 3 Na plus than in 2K plus. It creates an imbalance of charge across the membrane.
So it's not just about the chemicals, it's about the electricity, too.
Precisely. It generates an electrical potential difference, a voltage, across the membrane. We call this the membrane potential.
And why does the cell need that voltage?
Oh, it's fundamental. That stored electrical energy is what nerve cells and muscle cells use for signaling. Action potentials, the basis of nerve impulses, are basically rapid changes in this membrane potential. Without the constant work of the Na plus K plus pump setting up that gradient, our nervous system wouldn't function.
Wow. Okay, so the membrane structure directly enables something as complex as thought and movement. Amazing. Let's shift gears slightly. We've talked about transport through the membrane. What about how cells stick together to form tissues? That involves proteins too, right? Cell adhesion molecules or CAMs?
Yes, CAMs are crucial. They're often integral proteins whose external domains reach out and bind to CAMs on neighboring cells, or to the extracellular matrix, the glue or maybe the mortar holding tissues together.
You mentioned adherens junctions using cadherins acting like Velcro. But then there are tight junctions. They sound even more serious.
They are. Tight junctions form a really, really close seal between adjacent cells. Think of them less like Velcro and more like rivets or welding seams joining 2 sheets of metal. They create a waterproof barrier.
Waterproof. Where would that be essential?
The classic example is the blood-brain barrier, the BBB.
Of course. Protecting the brain.
Exactly. The cells lining the tiny blood vessels, the capillaries, in the brain are joined by extremely effective tight junctions. This prevents almost anything from just leaking out of the blood between the cells and into the sensitive brain tissue.
So anything getting into the brain has to go through the cells. controlled by transporters?
Pretty much, yes. It forces highly selective transport across the cells rather than allowing leakage between them. It's a critical protective mechanism, all thanks to those specialized membrane junctions. Shows how membrane structure dictates tissue function. Incredible layer of control.
Okay, moving from structure to communication. The cell needs to receive signals from the outside world, right? Hormones, growth factors. How does it get the message? That's the job of cell surface receptors, I assume.
Precisely. Receptors are the cell's antenna, or mailboxes. They're usually proteins embedded in the membrane with an outer part designed to bind a specific signaling molecule, or ligand.
Like insulin binding to its receptor.
Perfect example. When the ligand binds, the receptor protein changes its shape, particularly on the inside of the cell. That shape change is the signal. It kicks off a cascade of events within the cell.
And the insulin cascade is a fantastic illustration. Blood sugar is high, pancreas sends insulin. What happens at the membrane level?
Okay, so insulin binds to its specific receptor on, say, a muscle or fat cell. Now, this receptor is special. It's not just a receptor, it's also an enzyme, a tyrosine kinase.
A dual function protein.
Right. Binding insulin activates its enzyme function. It immediately starts adding phosphate groups taken from ATP onto itself. It autophosphorylates, and then it phosphorylates other proteins inside the cell.
So that phosphorylation is like flipping a series of switches, the start of the internal cascade.
Exactly. It's the trigger.
Yeah.
And that cascade rapidly leads to a very specific action. The cell mobilizes vesicles, little internal bubbles, that are storing pre-made GLUT4 glucose transporters.
So it's not making new ones from scratch. It has a stockpile ready to go.
It has a reserve fleet. These vesicles quickly move to the cell surface and fuse with the plasma membrane.
Like merging onto the highway.
Yeah. And this fusion process instantly inserts a whole bunch more GLUT4 transporters into the membrane. Suddenly, the cell has massively increased its capacity to take up glucose from the blood.
Wow, that's fast. A rapid deployment system triggered by the signal.
It's incredibly efficient. A direct, rapid restructuring of the membrane's transport machinery in response to an external command. Shows just how dynamic that membrane really is.
That adaptability is stunning. But none of this works if cells can't recognize each other, right? Especially for the immune system. How does a cell show its ID?
That's where the carbohydrates come in. On the outer surface of the membrane, you have this fuzzy layer called the glycocalyx.
Glycocalyx, okay.
It's made of short carbohydrate chains attached either to membrane proteins forming glycoproteins or to membrane lipids forming glycolipids.
Like a little sugar antenna.
Sort of, yeah.
Yeah.
And the specific patterns of these sugars are unique. They act as cell-specific identification markers. Your immune cells are constantly scanning these patterns.
To tell self from non-self.
Exactly. To recognize your own healthy cells versus a bacterium, a virus-infected cell, or even a cancer cell which might have altered glycoproteins. These carb patterns are the cell's ID card.
So wrapping this up, this deep dive really shows the membrane isn't just a simple wall. It's this incredibly complex fluid mosaic.
We're at a dynamic entity. You've got the phospholipid foundation, stabilized and organized by cholesterol and lipid rafts. You've got proteins doing everything from transport.
Channels, carriers, active pumps, setting up that vital membrane potential.
To adhesion, holding tissues together like at the blood-brain barrier, and acting as receptors.
Triggering those rapid internal responses, like mobilizing GLUT4.
And finally, the carbohydrate IDs for recognition. It's a strategically organized, almost, well, semi-sentient boundary.
Yeah.
Constantly working.
Yeah, it dictates so much how we get energy from glucose, how our nerves fire, how our immune system functions. It really is the ultimate smart boundary, balancing keeping things out with letting the right things in and communicating effectively.
It really is. And that dynamic nature we saw, especially with the insulin response, how the cell can literally fuse internal vesicles to rapidly rewire its surface composition, makes you wonder.
What's that?
Well, given that capability, that fluidity and constant restructuring,
Yeah.
what are the limits to how quickly a cell membrane can adapt? If faced with a totally new environmental challenge, a new toxin or stress, how fast can that fluid mosaic physically reorganize itself? Is there a fundamental speed limit to that adaptation?